another session. Just give me a few seconds to make sure we are live. Okay. I'm just waiting and checking if here it shows anything. Usually there's a time lag, so please be a little patient. Yeah, there you go. We are live, and let us see if there's any audibility issue. Okay, sounds good. So let me go ahead. Okay. So the topic for today is polymerase chain reaction. Okay, PCR. We call it in short. We call it as PCR. I'm your educator Nivedita, and I'll be taking up your session for now. Okay, today's biology session will be looked after by me. Okay, if you're here new to this channel, okay, we have Unacademy has many channels, obviously, because there are so many competitive exams as well as even school training that Unacademy provides. Okay, now here we are at KCET, so this is the KCET channel of Unacademy. Okay, so usually if you want to see my classes, if you like my classes and you want to watch them, I do special classes as well as plus classes at the at our regular platform on the app. Okay, for the link for the app must be given down at the description box. So you can go there and you can watch there. So special classes as well as plus classes, I do it on a regular basis on the app. So every day at least one or two classes of mine will definitely be there. Okay. So what you need to do is you can go down to the description box there, and at the bottom, the first thing that is provided, I think the first link is to my profile. So my profile at Unacademy, the app. Okay, so you can go there and you can follow me. So if you follow my profile, what it will do is every time I'm live, it will tell you whether there's live. So you don't need to wait for any notification from anywhere else. So the moment you see the notification, you can just click on that. It will take you to my class and you can go ahead and watch my class. Okay. So don't forget to do that as well. So I was telling you about, excuse me, I was telling you about uh, special and plus classes, right? So special classes are free and plus classes are subscribed version. Okay. So like what do you do? What we are doing right now, this is a YouTube session. YouTube sessions are more like a sneak peek. I'm going to give you obviously a concept coverage and MCQs here as well, but it's going to be crisp. It's going to be very small and neat. It's going to be that. If you are watching our sessions at the app, it is much more in depth. There is more concept coverage. There are more MCQs as well. So complete preparation, it will help you. Okay, amongst that also we have the plus classes, which are the subscribed version, which is much more helpful. Under that we have batch system, so we have multiple batches. So I take up for the comprehensive batch. Everything to do with biology for the comprehensive batch will be looked after by me. Okay, so don't forget that. Comprehensive. There are multiple batches. You can go for the comprehensive batch. So complete syllabus preparation, tips and tricks, topic-wise questions, full-length mocks. Okay. So if you become a plus member, don't forget to click on enroll in batch. If you don't do that, you will not get notifications when I'm live. Okay. So don't forget to do that. My code, I've put it up here. That is Nivedita Live. Okay, Nivedita Live. There's no H. Now, the monthly plan that we have is 2,250. Okay, now 2,250 is our trial offer, which is for a month. Instead, what you can do is, I usually suggest watch the special classes. If you like the special classes, which are free, then go for plus. Okay, so that is up to you. Okay, you can then go for plus, because obviously there are some topics which may not be covered in special classes. In plus, what we'll do is, for example, one chapter which is there in, for example, this biotechnology chapter, here I'm doing one part of it. special class, maybe I'll do like 1/4 or 3/4 of the chapter in one session. What I'll do it in plus is, in plus it's a course. So I will be doing 8 lessons on this chapter. So 8 sessions I'll be giving you, and with that so many MCQs as well. Okay, so you can go ahead and do that. So we have 6 months and 12 months, 3-month plan as well. You can choose whichever is the best way for you. If paying this amount may seem higher, remember you can go for EMIs. Okay, and also if you see these, as you increase the month, what happens is it becomes 1031 and 1688 per month. 2250 is different, and those, okay, so it reduces your amount. So 54% discount it gives every time your month plan, your duration increases. What do you get at plus? Premium content at affordable price, single subscription, unlimited access. So by this I mean PCMB, all subjects you're paying only once. You're not paying separately for each subject. Plus, if you're usually you get only one teacher or one educator to teach you, right? Here it is not like that. Here, any educator's class you can watch. Once you have a plus subscription, anybody's class you can go and watch. Okay. Structured live classes, I told you, break down sessions. Dedicated live doubt clearing session. Now, this is fun because a lot of questions from some simple questions to the most complex questions students can ask, which we clear out. Quizzes at regular intervals, depends on the educator. Okay, there are quite a few quizzes that I put up. Batches, I told you I take up for the comprehensive batch. Okay, test series, that is DPPs, that is daily practice problems, full length mocks are also there. I've been telling you about my code, Nivedita Live. The code is also given in the description box. So why I'm telling you about my code is, when you give a code, when you put an educator's code, that time it will show you all the discounts. You can see the discounts put up here. Plus an additional 10% discount is added. Okay, so you can do that as well. So you remember 17%. I told you, as you go, 3 months 17%, 6 months 25%. For 54 for 12 months and 67. So that as you increase the duration, the discount percentage increases. You can pay it through EMI as well. Okay, the links have been provided here and also at the bottom in the description box. So let me start off my session. Okay, so I will be talking to you today about polymerase chain reaction. Okay, my name is Nivedita and I'll be telling you about PCR. Before we go ahead, I'll just put up a blank slide so I can tell you more about PCR. Okay, let me find that slide. Yeah, there you go. So PCR, that is a polymerase chain reaction, PCR we call it as. It was first put up by a person called as Carey Mullis. Okay, it was first put up by a person called as Carey Mullis. Now here, what we do in PCR, what does the name say? Polymerase. Therefore, definitely that enzyme polymerase should be there, right? Chain reaction. Next statement says a chain reaction. So what do you mean by a chain reaction? There should be continuous reactions happening, one after the other, one after the other. So that also happens over here. When I'm explaining, you'll understand. Okay, so based on what is happening in the reaction, it is named that way. Polymerase chain reaction. That is, a polymer is being formed also, but the enzyme used is polymerase. Okay, so this is under this, why do we do PCR? The question is, why do we do PCR? When you have one particular gene and you want to increase its number, or you can simply say amplify. Amplify means what? Increasing in number. So you can, if you have one gene, it looks like oncogene, okay, one gene, okay, and you want to increase its number, like I said, you want to make multiple copies, you can make it, or you can also say clone a gene, or you can use the word clone. Okay, if you're trying to clone a gene, if you're trying to make multiple copies of a gene, that process, amplification of gene, is called as PCR. Okay, so how do we do it? I'll tell you more. So usually, if you see, let me go ahead to that slide and show you. So amplification of gene of interest, or GOI, we write it as GOI, gene of interest. So there's a particular gene that you're interested in. That's why it's called as gene of interest. Simple, right? Okay. So if you see here, this is the process. Okay, while looking at the schematic diagram, it may make no sense to you. Don't worry, I'll tell you more about it. So if you see here, there are three steps that I've mentioned: denaturation, annealing, and extension. So it is in a process of three steps. Okay, 3 plus 1, which is the last step, that is cooling, that is much more later. So three major steps which goes on repeating again and again, again and again. Okay, so that is denaturation, annealing, and extension. So from one copy of DNA, so you can see this one copy of DNA, multiple copies of DNA are done. So usually 1 billion copies of DNA you get. How do we do it? I'll just go to that empty slide and draw it and show it to you. Okay, because looking at the schematic diagram may feel a little daunting. So I'll draw it one by one and show you. So we have the first step I said, that is denaturation. What do you mean by denaturation? Denaturation majorly means breaking down, right? So denaturation. So imagine you have your DNA. Okay, obviously it would have two strands, 5 prime to 3 prime, okay, and 3 prime to 5 prime, and you would have hydrogen bonds between each of the what to say, nitrogenous bases. Okay, I'm showing it by this lines. So this is how your DNA is. Now imagine this to be your gene of interest. Okay, imagine this to be your gene of interest, GOI. Okay, now the first step is denaturation. Denaturation is where your gene of interest is there. Now you're going to break it into two separate strands. There are two strands are held together. You are going to break it into two separate strands. So that process is called as a denaturation. Now for that to happen, you need a good temperature. Okay, at least 90 degrees above. Usually 92 degrees you need. Usually 92 degrees at least you need. So what you do when you add that temperature, when you add heat to this, it will break up. Okay, your 5 prime to 3 prime and this one will separate up. So now the bonds have broken. Okay, can you see the bonds have broken merely by the effect of temperature? Merely by the effect of temperature. So this process is called as denaturation. If you remember, in protein also this happens. Its structure changes, we say, no? Here, because hydrogen bonds are easy to break, they break up. Okay. So this is how it happens. So this is the first step, that is denaturation, requires a temperature of 92 degree Celsius. Okay, for breaking down. So that's the first step. Let us go to the next step. Okay, I will clear this up. The next step is extension. No, sorry. Let me erase this. The next step is annealing. Sorry. It is annealing. So because for it to extend, we need to anneal something. What do you mean by the word annealing? Anneal means join. Okay, anneal means join. So what has happened now? We have your separate two DNA here, right? We have separate two strands now, 5 prime to 3 prime, because of denaturation they have separated. Now what I want is 2 to become 4, right? To become 4, so that I will have two separate gene of interest now. Okay, that is my goal is to increase in the number, so amplify. So what I'm going to do is, obviously, okay, you guys, I completely forgot to tell you, if you have anything to say, ask. You can put it up in the live chat. Yeah, I'm so sorry, the chat was not visible. I thought there was no chats. Anyways, hi Nityanand, and hi Ashik as well. Yes, it is making many copies of gene of interest. Okay, yeah, the chat is now visible. I had kept it on, but anyways, so the next step is annealing. Annealing, like I said, is fusing. So what do we do here is, we need to add something called as primer. So if you have studied replication, DNA replication, you realize that DNA just cannot start, your enzyme just cannot start what adding more another strand here. Instead, it needs a starter molecule. It has a starting problem. It needs a starter molecule. So that we call as primer here. Primers are nothing but oligonucleotides. What do you mean by oligo? Oligo means 2 to 9. Okay, usually 2 to 9. The word oligo means that. Nucleotides. So usually these are small chains of nucleotides. Let me change the color and tell you. So this would be your oligonucleotide. Okay, one oligonucleotide, another oligonucleotide. So these are we call them as primers. We call them as forward primer and reverse primer. Okay, depending on where they attach, we call them as forward primer and reverse primer. So what is our intention? Our intention is for it to join over here and join over here. Okay, now it has joined here and here. So our primer has now attached. So this is forward primer and reverse primer. Now for this attachment to take place, it requires a particular temperature. So that temperature usually depends on which primer it is, how big the size is, on all that. So generally around from 50, 55 to 75 ke beech mein. It will be somewhere in between that. Okay, but generally we tell 64 degrees Celsius. So we put 64 degrees Celsius. So 64 degrees Celsius temperature is required for this to join over here. Okay, now this joining, the process of joining means annealing. So this annealing process is done. Okay, annealing process. So this is the second step, right? The first step was denaturation, breaking of the bonds. Second step, the primers have attached. The primers have attached, that is annealing. So 64 degrees Celsius for this. Okay, let me move on to the next step. Okay, let me erase this and move on to the next step. The last step here is extension. Okay, it is extension. Now, extension means what? You're extending the molecule. Okay, you're extending the molecule. So now what has happened? We have our structure like this, right? This is how it has separated. Now, what we are going to do is 5' to 3'. Now, your primer also has attached, because of the previous this thing, we have attached the primer at each of these ends. Now, what we are trying to do is, we want them to extend. We want them to extend, right? So, what we do is, for the extension, we require DNA polymerase. Okay, we require DNA polymerase. I told you what is the reaction name? Polymerase chain reaction. So therefore, we require DNA polymerase. Now this DNA polymerase has to act so that this extends. Okay, so that this extends. Now for that to extend, what are the requirements? Don't you think we need some nucleotides so that we can add it? So in the solution, we already add some nucleotides. Okay, plus some usually ions like magnesium ion, which will help in polymerase to react. Now my question is, earlier on, this solution is already ready and put, okay. My question is, earlier on, we put a temperature of 92 degrees Celsius, then we put a temperature of 64 degrees Celsius. Don't you think, since DNA polymerase is an enzyme, don't you think it will denature itself? Don't you think it will get destroyed, right? Because our body temperature is 37 degrees Celsius, it cannot work beyond that, right? So therefore, it becomes a problem. We cannot use this polymerase because when we put it in that temperature, it will get destroyed. It will get denatured also. Okay, so that is why we use another DNA polymerase. We use a DNA polymerase that comes from a bacteria, that is Thermus aquaticus. Okay, that comes from a bacteria, Thermus aquaticus. Now, Thermus aquaticus is a bacteria that stays in hot springs. So therefore, its normal temperature is very high, right? So therefore, even at 92 degrees Celsius, it will be stable. So such a condition, we say it is as thermostable. That is, it is this enzyme is stable even in high temperature. So that is why we use this polymerase, and this polymerase we call it as Taq polymerase. Why we call it as Taq polymerase? It is a DNA polymerase only. We call it as Taq because T from Thermus, AQ from aquaticus. So Taq polymerase. Now when I add Taq polymerase, that can function. Now Taq polymerase, even though it can survive 92 degrees Celsius and lesser temperature also, it has its optimum temperature. Every enzyme has its optimum temperature. So the optimum temperature for Taq polymerase is 72 degrees Celsius. So when you put the temperature of 72 degrees Celsius, this will act very beautifully. So what it will do is, now with all the new DNTPs, it goes on adding. It will make a new chain. It will go on making a new chain here. Okay. So now Instead of one copies, don't you have two copies? Instead of one copy now, don't you have two copies? You started with one copy. Now you have reached two copies. So you can see how from denaturation to annealing to extension, two copies are made, right? Now what we do, we repeat the process. Remember I told you it is a chain reaction. What we do is we repeat this process. Again, we go to denaturation. So 2 now has become 4. Now 4 has to become 16, right? So it has to increase its number, right? So therefore, we are going to go to denaturation again. That is going to separate, new primer will attach, new this thing. It goes on happening. This cycle continues for around 30 times. 30 times. So when we do 30 times, it will have amplified. You will get at least 1 billion copies. Okay, you'd have gotten at least 1 billion copies. So this is how, this you can see the same process, denaturation, that is separation of the brands, annealing, your primers have annealed, then extension, that is your new DNA has formed. I hope you have understood this part. Okay, so usually these reactions, I will go back to the reaction once more. Usually this reaction we do in this tube. Okay, let me change the color. Yeah, we do it in this tube. Okay, now this tube is PCR tube, and you can see this is 2 ml. Okay, this is 2 ml. This is also 2 ml. This is very small compared to that. And usually, if you see only this part, only this much, what I marked here, only that much is your solution. Okay, only that much is your solution. So you can imagine at how much minute quantity your reaction is happening. Okay, this much region you have probably minute quantity of reaction. Now, to increase that temperature, reduce the temperature every time, we can't do it. That's why we have a machine, that is this machine, which we call as the thermocycler. Now, thermocycler, what it does, can you see these small spaces over here? Now, those are where you put these tubes. These tubes are put over here. Okay, and here there are numbers and everything. So we feed the time. Okay, denaturation step, so much time, so much temperature. Annealing step, so much time, so much temperature. Extension step, so much time, so much temperature. And then cycle again. So it has to do it 30 times till you get the final product. After the final product, I said there are 4 steps. The last step is cooling. So after everything is done, after the 30 cycles are done, we put it into cooling cycle. So at 4 degrees Celsius, it will stay, so that we can remove our product whenever we want. Okay, so that is how PCR works. So one rough going through of the steps, and then I'll go to MCQs. So polymerase chain reaction called as PCR. You can see your gene of interest. This is green in color now. We have denatured it at 92, 94, 96 degrees Celsius. And here we have added the second step, that is annealing. We have added our primers. Okay. Once we have added our primers, the third step, that is extension. You can see by Taq polymerase, extension happens, the blue one. So now you have 2 copies, and each of those copies will go on increasing and increasing in their number up to 30 cycles till you get 1 billion copies. So you have amplified your gene. From one gene, you have gotten 1 billion copies. So you have amplified your gene. Okay. So I hope you have understood PCR. It was a short, crisp session. Obviously, in depth, a little slower probably. I would have explained in my longer sessions. Anyways, okay, you can come and watch that as well, because in Plus I have explained much more in depth about this entire concept. Okay, let us go to MCQs. Again, there's a difference with our MCQs. You can answer here at the live chat. I will be watching. Okay, at the Plus or special classes on the app, I would give poll. So you just have to click on the answer. So let me start off. The first one, the polymerase enzyme used in PCR is DNA polymerase 1, Taq polymerase, reverse transcriptase, restriction endonuclease. What is the answer here? You can put it up in the live chat. Yes. Anything to say? Nobody? You guys are there, huh? I can see that you're there. Okay, no problem. In the poll, if you answer in our app, you don't get to realize who's answered and who's not. Here, you're worried that your answer is visible everywhere. No issues. Don't worry about that. That's the first thing to know. Doesn't matter. If you're right or wrong, you're learning right now. Okay, so here it is Taq polymerase, that is from Thermus aquaticus. Okay, next one. The first step in PCR is denaturation, primer extension, annealing, cooling, which is the first step in uh, PCR. You can go ahead and answer this one. I'll give you a few seconds. Okay, so the first step is denaturation, second step is annealing, third step is primer extension, and the fourth step is cooling. So therefore, it is denaturation. Okay, next one. In the figure given below, steps are there. So this is A, this is B, and this is C, and this is D. So what is A, B, C, D? Their options are given here. So one statement says that B is the denaturation at a temperature of about 98 degrees, separating the two strands. So is it B that way? Okay. Is B that way? No. B is not denaturation. B is primer annealing. So therefore that is not the option. Next statement says A, denaturation at the temperature of 50 degrees Celsius. You don't even need to see that because denaturation, it is correct, but 50 degrees Celsius, that is wrong. So therefore, that statement is wrong. Third one, C is extension in the presence of heat stable DNA polymerase. Yes, this is extension and it is, you have to use a heat stable polymerase. So therefore, this is the correct answer. A is not annealing, annealing is B. So therefore, this is also wrong. Next one. In PCR, which statement is incorrect regarding PCR? Okay. So in PCR, two primers are used. Taq polymerase is related for PCR. Taq DNA polymerase is not thermostable. Multiple copies of gene can be synthesized in PCR. So what do you think is the answer here, students? No idea. Okay. In PCR, it's true that we use two primers. Remember, I drew two primers to attach. So you have the forward primer and reverse primer. So this statement is correct. Taq polymerase is very important. It is related to PCR. Multiple copies are synthesized. That is also correct. DNA polymerase is thermostable. That is why we use Taq DNA polymerase. Otherwise, we can use any DNA polymerase. We use Taq DNA polymerase only because it is thermostable. So this not is the wrong part. Right? So therefore, this statement is incorrect. Next one. Which of the following is not necessary to execute a polymerase chain reaction successfully? All four DNA base pairs, short DNA base primers, DNA polymerase, DNA library. So what do you think the answer is here? Okay. So 4 DNA base pairs, ATGC or all required. Obviously, every given point of time they are always all required. So therefore that is true. Short primers also we require, right? The two primers that we add, the forward and reverse primer. DNA polymerase, specifically Taq polymerase, is very important. DNA library, however, we don't require. DNA library, we don't require. DNA library is where in your entire DNA, whatever is there, the information on all the DNA, this thing, and the structures and everything is not required. Which of the following is incorrectly matched? Agarose seaweeds, Thermus aquaticus tDNA, plasmid vector, SAL1, restriction endonuclease. Now, this question may be a little difficult for you to answer if you have not studied that biotechnology chapter. So, agarose gel electrophoresis is a process, okay, which we use to separate DNA. Now, yes, it uses the gel from seaweeds. Okay, plasmid DNA acts as a vector, that is to carry your gene of interest, that is a vehicle. SALH1 is a restriction endonuclease. It is a molecular scissor which will cut your DNA. Okay, so more on that probably I can put up a session. Thermus aquaticus does not give tDNA, it gives you Taq polymerase. Right? That is what we get from Thermus aquaticus. tDNA is something to do with another vector that we use, TI plasmid, in case of Agrobacterium tumefaciens for plants. So this is Thermus aquaticus. Which one of the following is a correct statement regarding DNA polymerase used in PCR? It is used to ligate, introduce DNA into recipient cell. It serves as a selectable marker. It is isolated from a virus. It remains active at high temperature, induced denaturation of double-stranded DNA. The question is, what is your DNA polymerase that is used in PCR? It is Taq polymerase. Correct? It is Taq polymerase. Therefore, this Taq polymerase is used for what? Because at high temperature, it should be able to withstand denaturation. So therefore, it is a D. That is the correct answer. It is isolated from a bacteria, not virus. Okay, selectable marker concept. I'm not sure if you're aware. It comes under a vector. Probably I'll put up a session on that. Okay, ligation also. DNA ligase can be used. Maybe another session on it. Now there are the different processes put up here and the temperatures that are given. So which temperature matches which process is your question. If you have time, you can answer this one. Usually I add extra time for such questions at the app, so you can directly watch each option and answer. So such questions usually, I told you, some questions in KCET require a little time, even NEET. You can give around 1 minute to such questions. So here, denaturation, it is 94 degrees Celsius, not 9 minus, sorry. It is 94 degrees Celsius. Annealing is around 64 or 60 here, maybe. And extension is 72 degrees Celsius. So the correct option here is A. Okay. Next, so I told you 92, 94, 90 is also correct. Annealing anywhere from 55 to 75 you can give. Extension, but is always 72. That number does not change. Extension is always 72. A biologist intends to use PCR, that is polymerase chain reaction, to perform a genetic task. The biologist is probably trying to discover new genes, clone a gene, cut DNA into many small fragments, isolate DNA from a living cell. So this question is indirectly asking you, what is PCR used for? Right? What is PCR used for? PCR is used for amplification. Right? It is used for amplification. That is making more number of copies, or we can also say cloning a gene, because the same gene is made again and again and again. You're repeating, right? So therefore, it is cloning of a gene. Last question. Processes are given here. A, B, C, and D is mentioned. So what is each one of them called? So the first one, if you guys are answering, I will wait. Otherwise, I'll go ahead. Okay. The first one is denaturation. Then we have annealing, and then we have extension, and finally amplified DNA, right? So what would be the correct option here? It is A, that is denaturation, annealing, extension, and amplification. So with that, my concept coverage and MCQs. This is a short session. I hope you understood PCR, at least basic understanding. Obviously, you can watch my session at the app also for better understanding. Okay. So if you like the session, don't forget to give us a thumbs up. That is a like button. Go and smash it. Okay. More for such classes, you can watch my special classes at the app. Also, you can join Plus. So if you join Plus, you can watch me at the comprehensive batch. So we have multiple batches. I take up for the comprehensive batch. So when you are joining, we have multiple plans. Introduction plan we have of 2,250. You can take a long-term plan also, whichever you are interested in. Okay, batches, I've already told you, we are live. My code is Nivedita Live, NIVEDITA LIVE. And when you use my code, don't forget to put it at the reference section, because it will show you all the different offers. Okay, all the different discounts will be provided. So you can go ahead and take up a subscription too. Otherwise, watch our free sessions, that is called as special classes. Okay, so I will see you at my next session. Okay, that is probably on the 16th, I think. Okay, till then, happy learning, stay safe, stay focused. Bye bye.